Welcome dear students. In previous video lecture we discussed on the physical processes that imbibition and diffusion. Now in this lecture we will going to discuss on the next process that is carried out during the absorption of water, that is osmosis. Osmosis is defined as the diffusion of water or solvent from a solution of lower concentration to the solution of higher concentration through semi-permeable membrane or plasma membrane, that is known as osmosis. The water at the interface of the cell wall and plasma membrane enters into the cytoplasm of the root hair due to the osmosis. Osmosis is the process in which the diffusion of water through lower concentration through the higher concentration through semi-permeable membrane, and due to that process osmosis the water enter into the cytoplasm of the root hairs. In respect or with respect to the concentration of that osmotic solution, three types of solutions are recognized or divides into three types that solution, that is hypotonic solution, hypertonic solution and isotonic solution. What is hypotonic solution? It is a weak solvent. It is a weak solution or strong solvent having low osmotic concentration. The solution which have low osmotic concentration is known as hypotonic solution, and that solution is a strong solvent is known as hypotonic solution. And then hypertonic solution, high osmotic concentration. The solution has the high osmotic concentration, and because of high osmotic concentration it is a weak solvent. Isotonic solution having such a concentration of solution where there is no gain or loss of water. Concentration outside and inside of the cell is the same. That type of solution is known as isotonic solution. According to the concentration or due to the concentration of solution, two types of osmosis is carried out: endoosmosis and exoosmosis. When the entry of water molecules into plant cell kept in a solution having lower solute concentration, then that cell sap is called endoosmosis, and because of that endoosmosis the cell become turgid. What is endoosmosis? When the lower solute concentration of cell sap, because of lower concentration of the cell sap, the water enters into the cell or into the cytoplasm of the cell. That type of osmosis is known as endoosmosis, and because of that excess amount of water is enter into the cell sap, and due to this the cells become turgid. Exoosmosis, the loss of water molecules from the plant cell, that is water molecules when out from the cell sap, then it is known as exoosmosis, and that happen because of higher solute concentration. And that type of exoosmosis, or because of exoosmosis the cell become flaccid. Endoosmosis causes turgidity, which increase the turgor pressure of the cell. Turgor pressure is the pressure exerted by turgid cell sap on the cell membrane and the cell wall. In a fully turgid cell, the DPD is zero, that water potential is zero in the fully turgid cell. The cell wall is thick and rigid and somewhat elastic. Because of turgidity, the cells have exerted pressure on the cell wall. The cell wall must exerted pressure on the cell sap, and that cell's pressure is known as wall pressure, and that wall pressure and turgid pressure are equal to same, but the direction of the that pressures are opposite. Osmotic pressure, the pressure exerted due to osmosis in the pressure exerted due to osmosis is known as osmotic pressure. Osmotic pressure is the pressure of the solution which is required in opposite direction so as to stop the entry of solvent molecules into the cell. Osmotic pressure. What is osmotic pressure? The pressure exerted due to the osmosis is known as osmotic pressure, and that osmotic pressure of the solution which is required in opposite direction so as to stop the entry of the solvent molecules into the cell. Therefore, diffusion pressure deficit is equal to OP minus TP. The TP is equal to wall pressure. So, dPd is equal to OP minus WP. That is, the equation is carried out. That is, Tp is equal to OP. In flaccid cell, the Tp is 0. The dPd is equal to OP. Osmotic pressure, that is, OP means osmotic pressure. And in the flaccid cell, the dP is 0. Then turgor pressure is equal to osmotic pressure. Facilitated diffusion. The passive absorption of solutes when mediated by a carrier is called as facilitated diffusion. The diffusion or the passive absorption of solutes is mediated by a carrier molecule, then it is known as facilitated diffusion. That particles that are lipid solubles can easily diffuse through the lipoproteinous cell membrane. The diffusion of hydrophilic solutes has to be facilitated because their diffusion across the membrane is difficult. Now next, the water potential. Every component of a system is having definite amount of free energy which is used to do work, that is the kinetic energy. Osmotic movement of water on the basis of free energy. That free energy per molecule in a chemical system is called as its a chemical potential. Every component of the system have definite amount of energy, or the molecules that are present have some kinetic energy or free energy, and that free energy per molecule of a chemical system is known as the chemical potential of the that substance or system. Chemical potential of water is called water potential. It is represented by the letter psi. Water potential of protoplasm is equal. Water potential of protoplasm is equal but opposite in sign to dPd. Diffusion pressure deficit, it has a negative value. The unit of measurement is as bars or pascals or atmosphere. Bars per pascal per atmosphere. Water potential of pure water is always 0. Water potential of pure water is always 0. Addition of any solute in it decreases its value. Therefore, it has negative value. Water always flow from less negative potential to more negative potential. That is from higher potential to low potential area. That are some factors that affect the water absorption. Now we can see that factors. Presence of capillary water is essential for absorption of water. The presence of capillary water is essential. When that water is not present, then the absorption is not possible. Next, the rate of water absorption is maximum at the soil temperature between 20 to 30 degrees centigrade. Soil temperature when 20 to 30 degree temperature, then degree Celsius, then the water potential is maximum. High concentration of solutes in soil water reduces the rate of absorption of water. High concentration of solutes in the soil water reduces the rate of absorption of water. Poorly aerated soil shows poor absorption rate. Increased transpiration accelerates the rate of absorption of water in irrigated soil. That is the some factors that affects the absorption process. Now one of the important processes also discussed, that is plasmolysis. Exoosmosis in a living cell when placed in hypertonic solution is called plasmolysis. What is plasmolysis? Exoosmosis in a living cell when placed in hypertonic solution is called plasmolysis. During plasmolysis, protoplast of the cell shrinks and reduces from or resides to the cell wall or periphery of the cell wall. Thus, cell become flaccid, that is plasmolysed cell is formed. Turgor pressure is always zero when such a cell is in a hypotonic solution. Endoosmosis occur, making the cell become turgid. This is called deplasmolysis in a fully turgid cell. When the cell placed in hypertonic solution, the water molecule present in the cytoplasm comes out the cell and cells become flaccid, and the cytoplasm or protoplasm present in the cell is going to periphery of the cell and large vacuole is formed. When that cell is placed in hypotonic solution, again the water enter into the cell and cell become turgid, that is known as deplasmolysis. Turgor pressure is equal to osmotic pressure, hence dPd is always zero. Then now we can stop here. In next period we can study the path of water in root.